I want to talk a little bit about how we have been using AI for spatial transcriptomics, but also in medicine. But most of all, I would like to give you examples of of how we here in Brazil perform low budget, high quality science. When I mean low budgets, it's kind of obvious because science is already per se low budget, right? So like we have been observing nature, and that's for free for thousands of years. But of course, there is a lot of cost in training people. So the university and research centers has to invest a lot of money for training the scientists. So if you are interested in looking things that are very, very far away, for example, when we create one single photo of a black hole, that was cost $55 million. And of course, and when you want to look things too close, the costs also increase. So if you have an experiment like this, it's gonna cost $23 million only for electricity for a year. And if you want to look many things at the same time, that is also be very costly. So one experiment can easily go to more than $10,000 per sample. So when I mean low budget, high quality science, I'm really talking about this part here. Once you already invest a lot of money in generating the data, in creating this repository where the data is available, and train the people. But these parts here are the examples that I want to show of things that we have been doing in the past years. And one thing that we focus is on precision medicine, which has to do with this. So imagine that you have a group of people that has the same disease. So they were infected by the same pathogen, or they received the same vaccine, or treated with the same drugs, and they have completely different outcomes. Some people are going to be fine, some people are going to be sick, and some people may even die. And that happens because every single one of us has a unique immune system. We have different age, transcription factors, stress, malnutrition, everything influences the outcome. So what we have been doing is to collect this huge amount of biological data, genes, transcripts, proteins, metabolites, and then we integrate this and try to find what is the minimal parts of this network that can actually predict an outcome. So this is the machine learning parts we have been using in our lab. So we apply this approach to several diseases such as chikungunya, Zika, pneumonia, influenza, Ebola, psychiatric disease, cancer, neurological disorders. So we have been using mostly data that were public available, but these four examples here, we actually paid for to generate the data. And of course, when COVID arrived, everyone was trying to study COVID, including us. We have been working in over 33 papers on COVID, but I'm only going to show you one unpublished paper on SARS-CoV-2 study. Jonathan, what he did was to look at transcriptomics data sets of SARS-CoV-2 infection. And the problem is that, come on, guys, there is already 56 transcriptomic papers on severe COVID. But the problem is that if you take two of these papers and then you look at the differentially expressed gene lists of reported in these two papers, you're going to see that the overlap is minimum, meaning that there is no consistency on this. So what Jonathan did was to collect all of these data sets, reanalyze them in an integrative way, and then we can, for example, look at not only one blood study, but we look at 8 blood transcriptomic studies. And then we try to find what are the genes, the signature that is consistent in these 8 blood studies. And then we apply the same meta-analysis approach for liver, kidney, colon, hearts, so far, so on. So we now know what are the robust signatures of each tissue in COVID-19. And then we integrate this into like a network where we connect genes and organs. And then we pick one gene to validate. So we pick RAB8B because RAB8B is from the RAB family that is known to be related to different virus, the phagocytosis of the virus and then the release of the virus. But there was nothing regarding RAB8B and SARS-CoV-2. So nobody knew about this gene in the context of SARS-CoV-2. So what we did was to validate this by infecting cells with or without RAB8B using CRISPR-Cas9. So if you have RAB8B, you have these viral loads, and if you remove RAB8B, the viral replication decreases, meaning that this gene was important for viral replication. We also create this dashboard where now you can just check the gene that you want. So you can look for the gene symbol, and then it's gonna tell you which organ is affected. So you don't need to reanalyze 56 transcriptomic studies. You can just check the gene that you want and study if it is changing in severe COVID. Single cell synthesis biology is another passion that we have been analyzed. So I'm going to report now another study where we use public available single cell data sets of brain metastases. So what Carlos was doing is Carlos was analyzing brain metastases that have different primary origins. So they came from melanoma, breast, lung, ovary, but all of them are brain metastases. So what we want to know is what are the shared molecular factors in brain metastases, regardless of the origin of the cancer. So he integrates all these data sets and then annotate the cell types, so he could see exactly what are the cells that were related to astrocytes, glia, nervous system, but these gray ones are the ones from the tumors. So it is obvious, but we can see clearly that the cells that were from breasts, they remain breast cells, tumor breast cells in the brain. Same thing with lung, melanoma, etc. So we can clearly see that they, even after integration, they are still bringing the primary tumor origins. Then we look at the composition of each cell clusters, and we try to find the communication between different cell types. So we use cell chat analysis that revealed that astrocyte was actually releasing a lot of ligands, especially to endothelial cells. And that is important because what we wanted to answer was what is driving the tumor cells leaving the blood and going to the brain, passing the blood-brain barrier. So we try to really understand what's going on during this passage of the tumor cells in the brain. So we use trajectory inference analysis, where we look at the transcription factors and the genes and the ligands over time, over the pseudo time. And then we validate this by looking at spatial transcriptomic data sets to try to see where the ligands and the receptors were physically located. And then we came up with these models that try to understand how the primary tumors release the tumor cell that goes to the bloodstream and then pass the blood-brain barrier, going to the brain, get established in the metastasis. Spatial transcriptomics, of course, is something very recent, and the idea is that you have an organ or a tissue where you can get an image from this tissue, and then you know exactly the position of XY coordinates of the cells. So you know in this image where the cells are. And then you can extract the RNA from the cells, and that's going to be the spatial single-cell transcriptomics dataset that we have been analyzing. One of the first question that we asked was in the brain, how the cells communicate to each other, if there were subsets of cells in the brain that are spatially located. And then we use a technology called MERFISH, which is an in situ hybridization kind of multiplex that allows you to see not only a single cell, this is the single cell, but also the single transcripts. Every single dot is a transcript colored by a different gene. Then this is already part of the paper where we have 3 sections of the mouse brain. And the first thing we did was to compare this data with what is known in the textbook. But the problem is that the textbooks has dozens of regions already mapped for the mouse brain. And if you try to merge this into real data, you're gonna see this problem. The red lines are the textbook areas of the mouse brain that is in the textbooks. But the gray dots are the real data. You see that it doesn't match. That means that we're going to say that this region is actually empty, but we know that it's not the case. And that's when we turn to astronomers, which has the same problem but with stars. So we reposition astronomer 2 to correct for this, and now we can see exactly what are the regions that are in our real data. And then we do the annotation, we identify the cell types of 700,000 single cells, and then we put back in this brain. And as you can see, neurons seems to have a very specific location in the brain. But not only this, if you actually get this neuron and recluster, so you put all the neurons again and then make a new clusters, you can identify 22 different subclusters based only on the expression profile. And these 22 clusters, when you put back in the image, look how beautiful it is. You can see that the neurons is clearly and specifically located in some regions of the brain, and we don't know why these subsets of neurons are there. But we also had to create novel spatial transcriptomic tools. So this one was inspired by the fact that most of the transcriptomic tools work like this. So you first annotate the cell type, you identify which cells are fibroblasts, which ones are macrophages, and then you put back into the image. That's not what we want to do. What we want is actually to see the biological process happening. For example, where inflammation is happening in the image, where type 1 interferon is happening, where autophagy is happening, regardless of the cell type. So we had to come up with a new tool called Spatial Enrichment Gene Set Analysis that has this logo here. So this is the lung cancer biopsy, a real lung cancer biopsy, and that's the spatial transcriptomic data when we create a heat map of 36 genes related to inflammation. This is supposed to show the areas that have inflammation in the cancer. But do you agree that it's very hard to see the boundaries of the cells? Where exactly is their place enriched? And then what we had the idea was to transform this enrichment into a height. And now we have a landscape of the cancer. And with that, we can use water to flood the noise. We can increase the water to get rid of the areas that are not inflamed. And then, of course, you can use the 3D printers to print the lung biopsies, which we did for some of the samples. So this is the islands of the same lung biopsy when you use exhausted T-cell genes, which are the cells that are tired of fighting cancer. And these are the islands that you create of the same sample, but now using genes related to effector CD8 T cells. And these are the islands of cancer, so you know where they are. And now you can navigate on these islands. You can see, for example, what are the composition of each island. You can see how much of this is fibroblasts, how much is monocytes, how much is tumors. But this is not the most interesting part to me. The most interesting part is actually to compare the islands. So if you have this 3D image, you can convert this into 2D image where the areas are the areas enriched for exhausted T cells, effector T cells, or cancer. And then you can compare the islands. For example, you can see the distance of each island of tumors compared to exhausted T cells. Same thing with tumors and then effector T cells. And that's how we know that a tumor island is closer to exhausted T cells islands than effector T cell islands. And then, of course, we are looking at different processes and to compare how distant or close they are in different contexts. And that became this tool called Archipelago, which makes more sense than Saga. And that is literally one of the computational tools we have been developing over the years. We are actually already in page 2, but I'm not going to talk about most of them. maybe I'm going to talk about one or two. This is how we can use archipelago on DNA barcoded antibodies, which is Codex, and that's a collaboration with Oxford. So Oxford people, they sent us the spatial transcriptomic data with markers of different stages of T cell differentiation. When T cells become single positive or CD4 or CD8 T cells. And then we create the archipelagos, the islands for each one of these cell types, the differentiation stages. And then we use gradient-based modeling, which is putting a ball on the top of the hill and then see where the ball is going to roll down. And then we use this to study how cells differentiate into the thymus. That's only a very small part of the image, just for you to understand. And these are the thymic epithelial cells that are probably playing a role in the differentiation of T cells. So what we did was to look at the islands that were formed by each one of these different types, cell stages, and then we can merge everything to see where the differentiation happens when cells became single positive CD4, CD8. Spatial is another tool that it's very new and has to do with this. Imagine that you have a lung cancer biopsy. Then you can do a spatial transcriptomic analysis where you identify the different cell types. What are the epithelial cells, CD4, naive, PDCs, etc. And what we are doing now is to including an epidemiological approach on top of this. Why epidemiological? Well, in epidemiology, it's very common to try to understand why, for example, there is a region with low burden of the disease surrounded by regions with high burden of the disease. So this is called low-high region. And similarly, why this city here or this region here have high burden of the disease surrounded by low burden of the disease. So this is the high-low region. And we have been applying the same thing for cancer and spatial transcriptomic data, why there is some regions that have low cancer surrounded by regions with a lot of cancer or vice versa. Why there is a place with a lot of cancer surrounded by places with no cancer. And vision, it's very easy to visualize this. You can actually map where the high, low, high, in low regions. And then we can see, for example, also in single cell, not only vision, but you can do the same thing with different cell types. This is the areas that are low, high, or high, low. And then the cell types that are here. And then you can count the frequency of each cell types on each one of these four regions. If it's high, low, high, or low, to see if there is any enrichment for a specific cell type. Not only this, you can look at the expression of the same cell type in a low-high region and high-low regions. That's going to tell you how activated this actually cell type is. This novel tool here, ImmunoVis, is a little bit different because we are merging computer vision with transcriptomics. And the idea is that neural networks is a very powerful machine learning tool that allows you, for example, to classify an image. So you can use neural network to tell that this is actually melanoma or not. The problem is that if you extract the RNA from the sample, and then now you have a table with 25,000 genes and hundreds of columns, which are different samples, and then you try to use the same neural network to classify these samples, you're going to fail. And you fail not because neural network is not good as a predictor, but it's because the biology is more complex than this. We may have cancer because of different combinations of markers of cancers. So it's very difficult to find very small sets of markers that is going to predict a very heterogeneous disease. But if you actually combine all these genes and put in the same image, so if you convert this information to an image, then you can use neural networks to classify the samples. And that's what we did. So we create an image, 2D image, where the dots are the pixels, are the genes, and the positions of the genes has to do with the expression profile how similar they behave in the same tissue, and how frequent they are present in different pathways. And then once you have this image, it's like you have a colored book, which is empty, and now you can color with the profile of the person. So when you have the image, the dots are not going to change anymore in the image, but the brightness is going to change if you have normal samples or cancer samples. Then you can train a model to learn from this image and then to predict a new image. And we did this, for example, for skin. This is the image of skin without coloring anything. And now you color for 3 samples of normal skin and 4 samples of skin of leishmaniasis patients, cutaneous leishmaniasis patient. Then you can clearly see that there is an image that is different. Same thing with health controls, now blood, and then with acute infection of chikungunya, which is very similar to dengue, yellow fever. And we did also for tuberculosis. So we took 4 independent tuberculosis studies where people have transcriptomics of active tuberculosis or latent tuberculosis. And for each sample, we create an image which we use to train the model. And then we use these independent data sets to test if the model was predicting right. This is to show that the samples that were active tuberculosis were predicted to be active tuberculosis. And the samples that were latent tuberculosis were predicted to be latent tuberculosis. Metatranscriptomics is another project where we are trying to investigate the microbiome of the people, which is Andres' job, but also the host transcriptomics, the integration of host transcriptomics with microbes, which is Icarus project. And the approach works like this. You have reads from RNA-seq, and the first thing people do is to map the reads into a human genome. And then the unmapped reads, they throw away. But what we want is actually to see if these unmapped reads are actually mapping to microbes. We are running this in almost 60,000 human samples, and I hope to show you some results soon. But I want to show one set of results coming from these studies here, the students here that were analyzing cervical cancer data sets. Cervical cancer is already known to be a slow progressing cancer that is associated with HPV infection. And what they did was to get bulk RNA-seq of these women with cervical cancer. And the authors already genotyped the HPVs from these samples. I'm only showing one bulk RNA-seq, but they actually analyzed 3 bulk RNA-seq, 1 proteomics, and 1 single cell of data of cervical cancer. And then I'm only showing 1 bulk RNA-seq. So here's to show that our metatranscriptomic tools can identify the bacteria and the virus. Now I'm showing the frequency of reads of each sample. Each sample is a different row, and the red bars are the HPV reads, and green bars are the bacteria reads. And these are the samples that were actually supposed to not have HPV, but we found HPV reads here. But when we look at the bacteria, that was surprising because we saw that some of the women were actually infected with Treponema pallidum, meaning that they had syphilis, and the authors did not know about that, and that could definitely impact the analysis, downstream analysis. But we also look at the association between certain bacteria and HPV levels, and we found that Cutibacterium acne, which is a very common bacteria in the skin, was associated with HPV levels. And this interesting bacteria is that is an important factor for establishing and maintenance of skin homeostasis. But because we also, so maybe this bacteria has to do with the slow progression of cancer. But because we also have the levels of HPV and the genes expression, we could compare the expression of the genes, each one of these 20,000 genes, and the HPV levels to see what are the genes in the host that were associated with HPV levels. And then we found 700 different genes that were positively correlated with HPV. And then we saw that most of the genes were related to cancer, cell proliferation, mitosis. But there were some genes related to Fanconi anemia pathway, which is a very interesting group of genes that has to do with cytogeneric instability, DNA repair. And that's awesome because these genes may be driving the slow progression of cancer. And in fact, authors already show that HPV can induce Fanconi anemia genes, and Fanconi anemia patients have more chances of developing cancer. But this study here was done in vitro, and this one was done in Fanconi anemia patients. And what they are showing here is in vivo in women that do not have anemia. And now I'm going to shift gears to talk about some of the projects on medicine. We have been using different narrow AI models for different purpose in medicine. Machine learning is the one that I showed you, but we are also doing single cell transcriptomics of leishmaniasis. And one of the messages here that I want to show is that bioinformatics can help you analyzing 20,000 genes and reduce the amount of genes to just few hundreds of genes. But after that, there is no other way of using bioinformatics to come up with insights. So when we create a figure like this showing the gamma delta T cells in cutaneous leishmaniasis, that was done with no program, but human expertise, conversation, looking, reading papers. There is no simple way of doing this. Cognitive computing has to do with the fact that we have been publishing too many papers. So in the past 2 years, 3 years, we have been publishing more than 1.5 million papers. That means that regardless of your field of expertise, you're facing a knowledge tsunami. You cannot keep up with the literature. Well, you cannot read 45 million papers, but a machine can read the papers and then can create these knowledge graphs where you connect genes, drugs, and disease. And then you can use the human insights to extract hypotheses from this knowledge graph. And that's what we have been using for drug repositioning of psychiatric and neurological disorders, also immunological disorders, also studying the evolution of knowledge over the 75 years, and a summary of what we scientists are studying over the 75 minutes in 1 minute for more than 500 diseases is shown here. So each bar shows the number of publications in that year for each one of these diseases. So everything here is in my LinkedIn, okay, guys? You can see my LinkedIn. Okay, so this is the evolution of how we are changing the focus of infectious disease to chronic disease, and then hepatitis, myocardial infarction. And then 85 appears HIV. HIV becomes the most studied disease. And then in the year 2000, obesity starts as a top disease study. Breast neoplasm was very much studied because of a lot of grants. And that cancer becomes a very much studied disease. And what happened in 2020? You see, guys, this is how we come up with a vaccine so fast, because everyone stopped what they are doing to focus on COVID. Well, when monkeypox arrived in 2022 in Brazil, I wanted to read all the papers of monkeypox. But the problem is that when I went to PubMed, there were 2,600 papers on monkeypox. I cannot even read the titles of 2,600 papers. But a machine can read the papers and then can organize into topics. And then I can put in a graph like this, where the articles related to neurological risk are here. The ones that are related to CD8s are here. The ones that are related to pregnant women are here. So meaning that if I want to study a specific topic, I don't need to read 2,600 papers or titles. And then you can see the authors that publish the most on each one of these topics, and that's how we know what are the experts of monkeypox. Computer vision, we have been using to diagnose and identify pathogens, parasites such as Trypanosoma cruzi or Leishmania, but we are also looking at skins to determine if the person has or not monkeypox. GUN is a project that we are trying to make a low-cost technology such as X-ray that have a low radiation exposure into a CT data, which is very expensive, but give you much more details of the sample. So this is the equivalent of have a 3D X-ray. And that was inspired by AIs that can get a 2D image of a face, human face, and transform into a 3D image. We want to do the same thing, to use generative adversary networks to transform X-rays into a CT image. And Mateus is actually has some very nice preliminary results showing that this is possible. This is another project where we try to look at distress syndrome of babies. So when babies have a distress of breathing, physicians evaluate if they had to intubate or use surfactants based only on vision and hearing. So there is no objective measures for quantifying how bad the baby is actually breathing. So we want to create an AI that captures video and sounds and then transforms this into a quantitative result that can allow us enable or assist physicians in their decision making. This was me breathing, and we are measuring how fast I was breathing. But then when my postdoc had a baby, he's filming his baby, and then we saw that we had to come up with innovative solutions to film the breath of the baby, and we now know what to do. LLM, we are using large language models for analyzing, for example, Mayo Clinic websites. Here you have 2,000 diseases where physicians wrote about the disease in the same way: overview, symptoms, treatment, cause, risk factors. And what we did was to convert this into embeddings, which are vectors with numbers for all the 2,000 human disease. And then we put back into a graph where the dots are the disease, and then the similarity of how they are described in the medical literature is based on the Mayo Clinic text. And this is to show that 3 diseases that affect somehow the lung cancer, chronic obstructive pulmonary disease, and asthma are very far away in this graph. And then you can actually see how close or distance are influenza, COVID, pneumonia, tuberculosis. And what we want to do here is a calculator of disease. What happens if you include fever in this disease? What happens if you remove arthritis from that disease? Which disease is the most similar one? This one has to do with one of the global threats, according to WHO, which is vaccine hesitancy. So we create a WhatsApp chatbot that can deal with fake news. And sorry, this is in Portuguese because it receives text, an image, and sounds, and then tells if this is true or not. So here I'm saying, is it true that drinking lemonade cure monkeypox? Oh, this is not true. There is no evidence for this. And then I create this image saying that monkeys that bite you can cause monkeypox. And then the bot says, oh, this is actually not true because monkeypox, I don't know why. Oh, sorry, guys. This is now when you will upload the image that I'm telling, is it true that if you get bitten by monkeys, you're gonna get monkeypox? And then tells you, no, this image is fake. This is not true. You cannot get monkeypox from this. And then you can actually record an audio saying like, oh, my uncle said that he got monkeypox from the bathroom. And then it says, no, this is not true. You cannot get monkeypox from the bathroom. And that's very cool because we scientists face a great challenge of replying to many fake news during the pandemics, right? So as a conclusion, Juliano is probably thinking, okay, sure, when AI is going to cure all the disease? And that's very hard to answer because humans are linear thinkers, meaning that if you take the human knowledge over the 150,000 years of human evolution from the it's very easy to predict what's going to happen during this time here. So if you are in medieval times, you can predict 5 generations from now. But if you are here, it's very hard to predict what's going to happen next year. But the only thing that I can say is that it's going to be fast. And exponential is very hard to predict. And one of the examples is the Human Genome Project that took 14 years and $2.7 billion to accomplish. In the first half of the Human Genome Project, only 1% of the genome was sequenced. In the second half, 99% of the genome was sequenced. So that's why it's so hard to predict something, a period that is actually exponential. But the fact is that there is a train named AI coming in a very fast speed, and we had to choose if we want to try to get on board or if we get distracted, think this train is never going to arrive, and it's going to arrive. And the conclusion here is that I showed you many examples of plenty of public available data sets that are there and bioinformatics tools that have been developed, which are user-friendly. I'm not talking about this, but you can also use ChatGPT, Claude to programming. But that does not mean that does not mean that you can get a good science just by applying this here, because of this book here. I don't know if you guys read the book Hitchhiker's Guide for the Galaxy, but they tell a story very nice of aliens that were tired of thinking about what is the meaning of life, the universe, and everything. So to answer this question, they built a computer of the size of a city called Deep Thoughts, and then they asked the computer, okay, what is the meaning of life, the universe, and everything? And then the computer said, I'm gonna have to think. Okay, so how long you're gonna have to think? 7.5 million years. Okay, 7.5 million years to answer this question? Yes. And then 7.5 million years later, the whole planet came to see the revelation that the computer has to give. And then when they turn on the computer, they ask, what is the meaning of life, the universe, and everything? And then it says 42. Say, what? How 42 can answer the meaning of life, the universe, and everything? And then the computer said, okay, I did this calculation many times, and the answer is 42. But maybe you did not know how to ask the right question, because the right question is going to give you the answer, which is 42. And then the alien says, okay, what is the question then that gives you the answer 42? And then the computer says, I'm going to have to think. And then the planet was destroyed before it gave you the answer. So the message here is that AI and superpowers, computers, will not matter if you don't know how to ask the right questions. So more than ever, we have to think more about questions now than answers, because AI is going to help you to answer the questions, but if you don't know how to ask the question, then there's nothing to do here. And with that, I'd like to acknowledge the team that did most of this work here. And of course, thank you all for your attention.